Good day, everybody. Thank you very much for the opportunity to talk to you today and to present this virtual presentation. I'm Iain Patterson from the Centre for Biological Control at Rhodes University in Makanda, right in the middle of the Thicket Biome. And today I'm going to be talking about some of the invasive alien plants that threaten and damage this very special biome. Alien invasive plants are a serious problem in South Africa. There are at least 773 alien plant species that have naturalized in the country, and 379 of these are regulated through the NEMBA regulations as declared weeds that are problematic or threatening in South Africa. And the thicket biome has a large number of alien invasive plants itself. In particular, the thicket biome is impacted by succulent invasives such as the cactaceae. A variety of creepers are also problematic in the thicket, such as Pereschia, which is the one shown in the photo on the left of the screen, and also other creepers like Andreda recordifolia or Madeira vine. There are also a number of tropical and subtropical shrubs such as lantana and inkberry that are problematic in the thicket biome. And then there are also the usual suspects that are problems in all biomes, such as the invasive alien waterweeds and a couple of alien invasive trees, particularly from Australia. Thankfully, there are many cases where biological control can help to reduce the negative impacts caused by these invasive alien plants. Before going any further, I'd like to start with a definition of what biological control really is, because there's often quite a lot of confusion around the term. My definition of biological control is the use of host-specific natural enemies to reduce the negative impacts of invasive alien plants. So I've got host-specific and reduced in bold, because those are key words within this definition. Host specificity means that the biological control agents should only feed on a limited number of plant species. And the plants that they feed on should be those that are acceptable to be fed on in the context of where they are released. So the biological control agents should ideally only feed on the target weed or close relatives of the target weed that are either also not considered valuable in the country where the agent is released, or are not present at all in the country where they're released, so they aren't a threat to those plants. So biological control agents should never feed on any indigenous plants or plants that are valued in any way. And I'll talk a little bit more about host specificity in the next slide. The word reduced is also in bold text, and that is just to emphasize what we're aiming for in biological control. We do not aim to eradicate these invasive alien plants. We aim to reduce their negative impacts and provide a long-term sustainable control at a landscape level. Biological control never will completely eradicate an alien invasive plant, so we can't see that as a goal of biological control. And this is very important in terms of understanding what we can expect from biological control and how we evaluate success. So the most important characteristic of a biological control agent is host specificity. And we determine the host specificity of biological control agents before we release them through a process called host range testing. This is a procedure that uses standardized protocols that we've used since the 1970s, although we are constantly always trying to improve how we assess host range. And we know from a variety of post hoc studies that this protocol can accurately assess the host range of biological control agents. And the fact that we can accurately assess the host range is also evident from the number of biocontrol agents that have been released worldwide. Over 400 have been released all around the world, and none of them have caused significant damage to plants that they were not expected to damage. So there have been some non-target impacts due to biological control, but all of those were predicted prior to release. In South Africa, we've released 136 biological control agents, none of which have fed on anything that we wouldn't have expected them to feed on. So we can accurately predict host range through standardized protocols of host specificity testing, and we can do this with a very high level of accuracy. The photograph that is shown in the slide is just what host specificity can look like in the field. This is a photograph taken at Hartbeespoort Dam. All of the brown is in fact water hyacinth that is being destroyed by Megamelus scutellarius, which is a biological control agent released for the control of water hyacinth. The plant is very brown because the agent is feeding on it, and there are millions and millions of biological control agents on that water hyacinth. The green plants are other species of plant that have grown on the mat of water hyacinth, and despite there being millions of the biological control agents right next to them, they have not been fed on by the biocontrol agent. So this is just a really nice illustration of host specificity in the field. In the next few slides, I'm going to illustrate what we can expect from biological control and how we quantify success and define success in biological control. And I'm going to use this schematic diagram that Hoffman and his colleagues published in 2019. On this axis, we have the extent of the invasion, which could be the distribution or the density of the alien invasive plant. And you can see that as the extent of the invasion increases, we get an exponential increase in the costs of the invasion. So those are the negative impacts associated with the alien invasive plant. And it's really important that at the high end of the extent of the invasion is where you get exponentially large invasion costs. The bottom axis is time. This could be measured in years, decades, or even centuries. And the line in the graph is the trajectory of the invasion. So at the point 0 here is when the alien invasive plant is introduced into its new environment, and it'll slowly start to grow with a bit of a lag phase at first until it starts growing exponentially. And at some point it'll fill up all the vacant niche available to it, and the line will asymptote. Mechanical control can reverse the trajectory of this invasion process, but at some point we're going to run out of money, or we are not going to be able to get back and do follow-ups indefinitely. We'll almost certainly not be able to drive this invasion all the way down to eradication unless we start at a very early stage in the invasion process where eradication might be possible. But in most cases, we are far too late when we start using mechanical and herbicidal control to control invasive alien plants. And at some point when we walk away, the trajectory of the invasion will go back to exactly where it was, and in the end, we're going to end up with just as much of the invasive alien plant as we would have before. So although we've saved quite a lot in terms of the invasion costs during this time period here, ultimately we're going to end up with just as damaging invasion as we would have even if we hadn't done any mechanical or chemical control. Biological control has a different impact on the trajectory of the invasion. If we initiate biological control at the same point as we initiated mechanical and herbicidal control in the last slide, you can see that we can change the trajectory of the invasion, and that this change is permanent. So we get a permanent savings in a reduction in the invasion costs. In this case, after the release of the biocontrol agent, there's still more of the alien invasive plant. The population continues to increase, but at a different rate. And even though this is not ideally what we would hope for from biological control, this should still be seen as a success because of the invasion costs that are saved in this scenario. We do, of course, hope for more than that from biological control. In many cases, we get a reversal in the trajectory of the invasion, and this will also result in a permanent reduction and a large savings in terms of the invasion costs. So the population of the biological control agent will be reduced below what we call a reversal threshold, so that there's less of the alien invasive plant than there was before biocontrol was released, and it will be kept at this level permanently. When complete biological control is achieved, this is when the trajectory of the invasion is reversed and is reduced to below some sort of tolerable threshold, where the alien invasive plant in question is reduced to a point that the invasion is rendered insignificant, and although the the plant will always still be in the system, it is at such a low level that it no longer has any real invasion costs. And so having the plants in the system has no negative impacts. And this is the best case scenario that we are looking for in biological control. Now, in the next few slides, I'm going to talk about some of the biological control programs on invasive alien plants in the thicket biomes. And I'd just like you to think about where you would place those biological control programs in terms of the level of success, whether you would place them in zone 1, 2, or 3, and how much of an invasion cost we have reduced due to biological control. As I've already mentioned, many of the most problematic invasive alien plants in the thicket biome are cactaceae. The cactaceae are largely endemic to the New World, so all cacti that are found outside of North, South, or Central America are in fact alien plants. There's only one species of cactus that's indigenous outside of that distribution. It is found in South Africa, and it's a small species of cactus that grows like a mistletoe or a lithophyte, and is very easily distinguishable from other cactus species in that it doesn't have any thorns. But the cactus in this photograph is one that everyone here will probably know. It's a Ficus indica, and it's so abundant that it could even be considered a keystone species in the thicket biome. It's really a noticeable plant, and it is really very, very abundant. It's from Mexico, and was introduced into South Africa many, many years ago because it's very useful for its fruit, which are really delicious to eat, and also the spinous varieties are very good for fodder. But the spiny cactus became extremely invasive in South Africa after it was introduced, and became very problematic to farmers, blocking up farms, stopping livestock and wildlife from moving through grazing areas, and also stopping access of livestock and wildlife to shade and water sources. So it started having a serious negative impact on both agriculture and on indigenous biodiversity. The two biological control agents that have been released for the control of Apuntia ficus indica are the Cactoblastis cactorum moth, which is this wonderful moth that was released in 1933. You can see in the photograph at the bottom here that this is an adult moth laying its egg stick, which it attaches to the end of a spine of a cactus, and the eggs are stacked on top of each other, and they hatch from the top down. The larvae crawl down the spine and then develop within the cladode or pad of the cactus, and you can see the beautiful orange larvae feeding inside of the cactus there. Although it is a wonderful insect, it has done very little in terms of controlling this alien invasive plant in South Africa. It's widespread, you find it quite often, but it is not particularly damaging on a landscape scale. The cochineal insect in the photograph on the right-hand side, your right-hand side of the slide, was released in 1938, and it is actually far more damaging than Cactoblastis cactorum and has done a lot more in terms of control, although it is less charismatic, so it's got quite a lot less attention. So there are still parts of South Africa where Apuntia ficus indica most certainly could be considered problematic. But we estimate that there is 90% less of this plant in South Africa due to the cochineal insect. Here's a photograph taken in the Karoo, not in the thicket biome. The first one is in the 1930s, and the next one in the 1960s, after biological control had been at work. And then the maps tell the story even better than the photographs. In these maps, the area in red was heavily infested, yellow is medium infestation, and lightly infested areas are in green. And you can see that in the 1930s, the area that it covered was much larger, and the extent of the heavy infestation was much bigger than it is today. So since the 1960s, it has been reduced to the point of the map at the bottom right. You can see that in our thicket biome, it is still quite abundant in places, but overall there's been a huge reduction, and this has certainly benefited South Africa as a whole. In the thicket biome, with the really dense vegetation that we have, the crawlers, which are the dispersal stage of the cochineal, really struggle to get from plant to plant, and that, as well as quite wet, cold winters at times, has reduced the efficacy of the biological control agents in this biome. But there are still hardly ever very dense infestations, and the plant is also of value, and people use it to collect its fruit. And now that it's been reduced to less dense infestations, people can actually get in between the plants to get at the fruit. So I believe that biological control has reduced this invasion to the point that we can now receive benefits from it, and it is much less damaging than it was. Another problematic invasive alien cactus in the thicket biome is Cereus jamaicaru, or the queen of the night cactus. You can see in the photograph on your left, the plant is absolutely covered with fruit, and every one of those fruit is packed with seeds. And that's the primary mode of reproduction of this alien invasive plant. The photograph on the right is covered in galls. Those are the galls of a biological control agent. It's a species of mealybug, and it forms these fluffy gall-like structures on the plant, and you'll notice that that plant has no fruit on it. So because the biological control agent stops plants from fruiting, there is very little recruitment of new plants into invasive populations. So the top three graphs are of populations without the biological control agent. You can see that there are very high numbers of young plants in all three of those populations, and this indicates an invasive population that is growing. So with lots of young plants, you're always going to get more and more older plants. The three graphs at the bottom are where the biological control agent has been established. You can see that there's very little young plants in the system, and these graphs indicate that the populations with the biological control agents are in decline, whereas those without the biological control agent are growing and the invasion is increasing. We have also done post-release evaluations where we've actually gone to sites where the agent was not present, released them, and monitored this over time. We've done this in 3 different sites, one in the Groot Marico area, one up in the northern KwaZulu-Natal, and then one around Makanda in the Thicket Biome. And you can see the first releases were done in 2016, and at all sites there's a steady decrease in the density of the plants until they are very, very low levels. So this confirms that within between 6 and 8 years, all invasive populations of Queen of the Night cactus should be under complete control from biological control, as long as the biocontrol agent is released at that site. This is what the impact of the agent looks like in the field. First small plants are killed, and then eventually even large plants succumb to the pressure of the biocontrol agent and die. Some cacti are much more difficult to control than others, and one that is really difficult to control is this jointed cactus, or Apantia aurantiaca, another species that is very problematic in the thicket biome. This is an extremely dense infestation, and you can just see how this would negatively impact any animals, including people, who would want to move through this land. It's a really spiky, horrible plant with huge negative impacts to agriculture. The biological control agent is a different species of cochineal to the one that's used for the control of Apantia ficus indica. In fact, the 2 different cochineal species will not even feed on the other host plant. So this is Dactylopius ostrinus, and it only feeds on Apantia aurantiaca. If you put it onto Ficus indica, it will not survive. And you can see from the photograph what the females look like. The tiny little red spots are actually the mobile stage of the cochineal, called the crawlers, and then the dead cladode below is one that the cochineal has sucked dry. We mass rear and release this biological control agent in very large numbers at as many farms as possible within the Eastern Cape area. And this is just a summary of a couple of sites where we've done post-release monitoring. And you can see the dark line is the number of cladodes that are the percentage of cladodes that are infested with cochineal at the sites. So you can see it increases after our release. And then after a period of about 2 years, you can see there's a steady decrease in the number of cladodes at those sites due to the impact of the cochineal insect. So we definitely get some success using this biological control agent against jointed cactus. But if we look more closely, we can see that the biological control agent is more successful at some sites than other. In this case, the dotted line is the amount of cactus that was at the site at the time of release. So at all 8 of the sites that are included in this analysis, you can see that there's been a decrease in the amount of jointed cactus that's at the site since our release. But you can see that for some of the sites this is temporary and there's a resurgence in the cactus. This could indicate that we need to release the biocontrol agent again, but it also shows that the control of jointed cactus is largely dependent upon the climatic conditions at each site. It is not as effective in shade and therefore not quite as effective in the thicket biome as it is in more open biomes such as in the Karoo. But with inundative releases we can get good control of this plant and reduce its negative impacts. Moving away from the Cactaceae, there are several other plants that we have successful biological control agents on that are problematic in the thicket biome. This is Acacia mearnsii, or black wattle, and in this case we use a biological control agent that reduces seed set. If you look at everything on the outside of that line that I've just drawn across the slide, you will see galls of the Dicynura midge, which lays its eggs inside of the flowers, and instead of them developing into seeds, they develop into these sterile galls that have 6 or 10 little larvae of the midge in them. And you can see that since the biocontrol agent was released at this site, no new seeds have been developed. All of the seeds on the inside of that line are from before the biocontrol agent was released, and after the release, all of the new growth is covered in galls rather than in seeds. And this reduction in seed set in the long term is reducing invasive populations of Acacia mearnsii in South Africa. There's a very long-lived seed bank, and it will take many, many years. But sterilizing the plants and combining this with some mechanical and chemical control will eventually result in a decline in Acacia mearnsii in South Africa. And hopefully soon we'll be able to say the same for lantana. This is one of the worst invasive alien plants in the country and is a really big problem in the thicket biome. But we have introduced an area-freed mite that forms these leafy galls where the flowers should form and stops it from producing seeds. It's not quite as effective as reducing the number of seeds as the Dicynura midges are on acacias, but it definitely reduces the seed set of the plant. Unfortunately, lantana is a really complicated species with loads of different flower forms, and the area-freed mite can taste the difference, so it only feeds on some varieties of the plant and not all of them. But there's definitely hope for the future, and hopefully we will be able to reduce the negative impacts of this terrible invasive alien plant using biological control. My last example of a biocontrol agent in the thicket is a waterweed. This is a photograph taken at the Blowkrans pool, which is in a very special part of the thicket biome between Makanda and Port Alfred. It's also the only breeding site of a minnow called Sandelia bainsii, a little fish species. And it is absolutely covered with the alien invasive Azolla folliculoides from South America. This is a very thick mat of this alien invasive plant, over 1 foot thick. I've never seen a mat of Azolla that thick because by the time I was working as an invasion biologist, the biocontrol agent had already been released. And the reason why I'm including this example is just to illustrate the sort of benefits in terms of the reduced invasion costs that biological control can result in. After the release of the Stenopalmus weevil, the mat of Azolla has been permanently removed from the Blowkrans pool, as well as nearly all bodies of water that were invaded by this species in the whole of South Africa. And the benefits from this biological control program have been absolutely huge. This is a very important sacred cultural site that can now be utilized again because the mat of Azolla is cleared. That critically endangered fish that I've talked about has open water that it can continue to reproduce in and may have been saved from extinction due to the biological control program. And there are very good cost-benefit ratios that have been calculated, which increase over time. Back in 2010, we got 15 Rand back for every 1 Rand that went into the program. By now, it's probably about 35 Rand. So the benefits from this biocontrol program have really been exceptional for South Africa and the thicket biome. And with that, I'd like to thank you very much for your attention. I am away at the moment, but I hope that I can get any questions that you have on WhatsApp. If not, I'll respond to you as soon as I can when I get into some reception. Thank you very much for inviting me to give the talk, and I hope that you enjoyed it. Thank you.